Thank you very much. I'm very excited to get the opportunity to speak to you guys. So today I'm going to talk to you with my professor hat as a professor at Northwestern, but I also do drug development. I've had the opportunity to take 7 drugs into clinical trials, and I'm hoping to take an 8th drug, which I developed based on ultrasonic vocalizations. All of my intellectual property is controlled by my university and our 2 startup biotechs. So first, let's talk about the problem of depression. This is something that you learn by doing clinical trials. So on the left-hand side is a pivotal study for probably the most recently approved antidepressant done by Sage Therapeutics, widely regarded as the best people in the business. They have the best clinical trials and get the best signals. And let's look at this data. So on the y-axis, Hamilton Depression Rating Scale. So the higher the number, you're depressed. And by the time you get to 14 down here, you're no longer depressed. So placebo versus their GABAergic drug, zuranolone. So what you find is on baseline, people are depressed. And then by the time you get to 15 days after treatment or after the start of the study, everyone's back to normal, right? And there is a small drug effect here across this first few weeks, showing an antidepressant response to the drug. So this is the hard data that those of us who do translational science have to deal with. So really what this says is that nearly everybody is cured after 14 days of placebo, right? This is shocking. If you haven't seen this data, it should shock you. And then, what's going to happen to the field? Since I'm wearing the professor hat, I get to be an eccentric professor. So please tolerate my literary references. It's a lot like Magic Mountain, where the depressed person, I'm going to be flippant because that's how people perceive people in these trials. Your dog dies, you're sad. You go up to Magic Mountain, you take some hallucinogens, you have a cosmic experience, you come down Magic Mountain, you get another dog, right? So this is a problem. This is going to sink the field from every perspective. One, this does not represent the disease. This is not respectful to the patients, the caregivers, and then the payers are just going to say, oh, well, we can give people Prozac. That's off patent. You can get placebo with that. And then more recently, people have done very evasive techniques with putting electrical brain stimulation, putting electrodes in people's heads, and then to cure depression. And then what happens when they find out that that's placebo, right? You don't really want to do that with some invasive stuff. So now we've talked about the problem. Here's a solution. So I'm a big fan of big data. I've been able to access probably 10,000, 20,000 data points from individual patients. And my favorite is the STAR-D study. So this is the largest antidepressant study ever run. It was an epic failure. By that, I mean, any drug you took in any order, you couldn't really predict treatment outcome. But what you could, if you dig in, you could see at the end of the study, after you went through 3 different antidepressants, are you still depressed, right? So there's a group here that are still depressed. These are the people with the most needs. Let's look at what their demographics were. They're older, but here, and I think this is the translational part that we're going to reverse translate. We're going to help the clinicians to actually do a better job measuring. They have more mental insomnia and less energy. And that seems to be a really consistent theme. And then they also show this groggy wake EEG pattern which I've also shown recently published is related to negative affect, physiological negative affect. And this can do a good job of sorting responder versus non-responder. So the real depressed people, the stable depressed people, there's something wrong with their brain. Their brain doesn't look right. It looks abnormal. And they have objective physiological measures of depression. What I'm not showing, and there's a major disappointment across these studies, subjective self-reports of emotion outside of energy don't correlate at all with treatment outcome or really disease very much. So let me take that EEG biomarker. EEG has been around for 100 years, but a benefit of it is you can look at thousands of patients across multiple disorders. So this alpha delta ratio, think of this as awake EEG. The more alpha you have, the more awake you are, the more delta you have, the more asleep you are. So you can look across patients with psychiatric disorders, depression, OCD, ADHD. They have a moderate suppression of this. But then when you get into neurological diseases, Parkinson's, TBI, Alzheimer's disease, it's very, very suppressed. We can capture this in animal models as well. And this isn't just a correlational fun thing. If you don't have any alpha and you're in a coma, that can lead to an end of life decision. So these things are vital for life. So synaptic plasticity, how does it work in the brain? What's going on with alpha and delta? I didn't pick these up sort of willy-nilly. These all came from synaptic plasticity studies. How do you induce synaptic dysfunction or long-term depression? You play a delta train to the brain. What happens during sleep? You get delta, and you forget things during sleep. Or many clinicians use delta to measure depth of anesthesia. Alpha, I call alpha sort of broadly from 6 to 10 hertz. This is enhanced synaptic function. This is how you induce long-term potentiation. You play alpha to the brain. So what we've shown, you get these faster waves, and then you get increased synaptic activity. I'm very much wanting to get into single neuron activity in freely behaving animals, but I was able to do similar experiments in primary cultures. And what these really are showing is that these fast rhythms are related to excitatory neurotransmission and activation of excitatory neurons, and the delta activity is related to inhibitory activity in these excitatory neurons. So let's help the psychiatrists out. Psychiatry really needs some ground truth, so let's go find some for them. So out of all the big data sets, the one that I like the most, I call the retirement party, or the retirement party study. So you go to a retirement party, you talk to the person that's retiring, and then 10 years later, let's see how they're doing. So the best predictors of these things, besides, I mean, there's things we all know. Exercise is important, but the two really strong predictors were wake at night. When at night you should be sleeping, the people with the poor outcomes are awake, so they have poor sleep, and they have a lack of energy and greater somatic pain. And I just put, just for comparison, the closest I could come up with a subjective self-report measure can be cheered up when down. There's a small effect, but it's very, very minor. So we're really capturing this other part of depression, not the acute, something bad happened to me and I recovered from it, like my flippant example of your dog dies, you get another dog, but a chronic long-term disease. So age is also a really good predictor, and it's something we can do translationally. I would say out of all these studies, age is probably the strongest, if not the strongest predictor. So many of these things you should know. Over time, you have less activity. Over age, as you age, you have less activity, you have less sleep. But here's kind of an interesting one I think is relevant for vocalization. So the young people get up, and then they can even become more active during the day. Whereas the aging brain, right around 5 or 6 o'clock at night, they start to want to have an early dinner and go to bed. So I think that that's something that we can measure in our animals. So here's sort of our original model for how emotions work in the daily cycle of emotions. So usually within a play bout, you can certainly see this with play, the animals start to chase and have these trill calls. And then usually when they're touching, usually the step calls in my experience are when the animals are touching each other, although that's not 100% the case. And then animals with good social skills will stop them. Animals with poor social skills will keep on playing until the other animal has to tell them to stop. And make the aversive calls. But then at the end, the animal sleeps and it resets, right? So here I'm doing 24-hour home cage recordings. I want to get into these more ethologically relevant visible burrow type systems, and I will when I get back. But these are long 24-hour recordings of the animals. And what we see here is in habituated animals, when the animals are supposed to be asleep, they're not vocalizing much. I think this data is maybe, as I collect more, it might not be this clean, but what I've shown is in the first hour or two after the lights go off, not immediately after the lights go off, maybe 15 minutes later, the animals show an incredible rate of vocalizations, and which goes down over time. And then this pattern also is captured in locomotor activity. And what you see with age is you see a decrease. I believe this is maybe a little extreme, and I believe in female animals you won't see this, but you see this pattern is also reflected in locomotor activity in the previous two slides. So the young brain sleeps during the night, their subjective night, and is active during the day, whereas the aging brain, or the brain with a neurological problem, is not able to completely sleep at night and isn't really all that active during the day. And we can actually capture this with EEG. But there's hope. You can take these older animals, and with play therapy, you can bring them back. So we recently published a paper showing the relationship between these EEG rhythms and emotional states in both rats and in humans. So I'll show you a video of the fast rhythm, the alpha rhythm. Hopefully it works. So exploring, sniffing, and the animal is sniffing and they're vocalizing. You can't hear the 50 kilohertz vocalizations, but this is an active, engaged animal. And sort of this, and the behavior, the sniffing, the vocalizations, and the locomotor activity and the brain activity are all coherent, right? And then in a human data set, so I can say that this was associated with positive affective vocalizations, and then I found a human data set with self-reported positive affect and was able to show the same thing. And then in contrast, you have the delta stimulation. Again, this stimulation is what induces LTP and what induces active wake behavior. And next we have the aversive call. Hopefully, luckily it will truncate, so we won't have to hear the whole thing. So the animal is rhythmically entraining its vocalizations to the optogenetic stimulation. I use optogenetic stimulation here. Electrical brain stimulation is actually much better. So you have a slow rhythm, a sleep rhythm that's coordinating these vocalizations. And this roughly relates to the intercall interval and the call duration of rodent vocalizations. So this teaches us a little bit about the psychophysics of positive and negative affect. Again, another literary reference. This best came from Thomas Pynchon and Gravity's Rainbow, that positive affect is Gaussian. All the good things in life are Gaussian. So it's well-behaved. For example, a good comic can make you laugh most of the time, whereas negative affect has a much higher threshold. These aversive calls, very hard to elicit, and they're unpredictable. And I will butcher this because I don't speak French. I call this a Poisson distribution. Hopefully that's close enough. A sad movie will make people cry sometimes, but not all the times. So, and this really follows synaptic strength. LTP and the ability to use LTP, alpha stimulation changes in synaptic strength, is pretty Gaussian, and you can increase it and decrease it and will regress to the mean, whereas large depotentiation leads to LTD, which will eventually break the connection. So it behaves very erratically. So this is kind of the most important part for translation. I'm building the building blocks for emotion, and how can I use them in their translational measures? So it's somewhat simplified, but I think this is also supported by the human literature of positive affect. Positive affect, both in animals and humans, is mostly social. The lion's share of it is social. So you have animals that are energized and they're doing what the circadian rhythm people would call right time behavior. They're active during their active phase. And at night, they're sleeping. They're not active. Whereas pathophysiology is you have low energy during the day and low energy during the night, and you're kind of having this groggy state. And you can capture this with lots of different ambulatory devices. And I brought my favorite, which is the device by Dream, which can capture EEG in an ambulatory way. This is the future, and we need to be reverse engineering to this. If this is how we're going to measure emotions and how we're going to do clinical trials, then this is how we need to support the clinical trials. So all of these things can be measured. With these devices, you can get EEG, you can get actigraphy to sort of get at your battery. You can also, at least in the US, you can take sound recordings of people and use them for clinical trials. So we can capture that. So what we've learned is that affect really is a vigorous social interaction, and valiance is this is really dependent not so much on the external stimuli, but your internal state. There's always two animals in the cage. Doesn't really change. Those two animals don't change, but the internal state drives what kind of vocalization you're going to get. So I'm trying to be also flippant. Feel like crap, take a nap ASAP, right? This is what we've learned from this, that negative affects is either the body's way of saying, take a break, or have a medical device. So I'm trying to develop a medical device for this that I can predict. So a useful way of looking at depression is not, I'm sad, what should I do? But more a chronic brain fatigue. So I think some of us can relate to the feeling of being jet lagged and lost in a foreign country. And many people have the story of trying to go to a train station and getting on the wrong train and trying to find your way to the conference. So yeah, I think that's a really a better way of thinking about it. A chronic brain fog and fatigue. The depressed state is more of, my brain's not working, I need to sleep, and my body hurts. I'm not sharp and attentive. And my favorite reference to Stanislav Lem, his master's voice. You know, I think it's the same sort of idea that you're not processing the information. You get all this information from a superior alien intelligence, and you're too stupid to understand it, right? And that is the beauty of Lem. And I think that this is also what's happening with these people. They're having a difficult time, and they can't navigate their world, and they can't solve these problems. And some of it is socioeconomic, and those are the people that really need the help, and we can help them. So next step, how do you make a drug associated with positive affect? We did one. It's actually rather simple. You take animals, you induce positive affect with tickling, you have negative controls, you do some discovery biochemistry, a molecule comes out. We're pharmacologists. We know how to take a big molecule, make it a small molecule, and then tweak it so that it gets across the blood-brain barrier. You find its receptor, you do receptor pharmacology, yada, yada, yada. Okay, so what does this positive affective drug do? So to quote my great mentor and friend, Yak, they are the fertilizers for the brain. This is what makes the brain grow. And here's the effect of our compound on spines in primary cultures. You can see where the arrow is. We make the spines grow. And this is the functional effect. We can get this also with electrophysiology. But this is really this is going to facilitate synaptic plasticity and learning. And then really, and I'm ending here, this is a translational endpoint. So what did we use with vocalization? What did we learn? We now know which patients we can dose. The people that have a problem with synaptic plasticity. We now know how to dose the drug. This was a real major issue in psychiatry. Most psychiatrists just want to dose the highest possible every day. Not how you dose these drugs. That's not how ketamine is dosed. So we know how to dose this drug because we can monitor synaptic plasticity throughout the brain. And now we know what to measure for efficacy, and we can inform the clinicians. I also go to conferences on positive affect, and the panel session that we're about to go to always agrees self-report's no good. You need objective physiology, physiological measures. But what we can do is we can help build self-report measures and other physiological measures that they can use. Remember my first slide, you know, the people at Sage were trying their absolute best to reduce the placebo effect. And that's as good as they can do. So we need these new measures. And that's how our data is going to inform. So we did doing basic science on emotion, and then we're able to create therapeutics and diagnostics, but also help to make the clinical trial better and help to have the patients, help to identify the patients that are in real need and help them. And that's my talk. Thank you for your attention.
Thank you. And now is time for questions. Okay, do you have questions? Yeah, so come here. I will have one.
I like repeat this from my talk at the previous conference, because it's like it's an interesting stuff, but like you can mathematically prove that by lone thinking you cannot create new information. You can only process what you already have. So this like shows why we need social affect or even like interaction with the world to create information. So this is why it doesn't even make sense to grow brain if there is no stimulation. Like there's no information. Like brain cannot self-stimulate to create anything new. And that's like the roots for what you're observing. So that was a comment. So I'm not asking anything.
No, that's good. And I think related to that is what we're finding is we're taking the vocalization to be more related to proper brain health. If your animal's healthy, Yak would say this all the time. If the animal is well fed and isn't scared, and you put him in a cage with another animal, they'll play. Right? This is the natural state of the organism, is to have positive affect. And I think I'm taking positive affect as well, but I'm also taking this and thinking about it in terms of brain health. Right? These same signals are seen in other neurological disorders. You know, that could use some brain fertilizer. And actually, depression at face value only has some of that, but we can enrich for these patients, and they're going to have more of a neurological problem. Also, you know, another exciting thing for me is this is another big data set that can be mined. I've only mined some of the nuggets from it. There's a lot more to be seen.
Right. So there is a question on Zoom. Which brain area or cell type did you stimulate during optogenetics?
Sure. So everything that I was doing was in the medial prefrontal cortex, sort of broadly, because I'm using this translational. And I'm also comparing electrodes that are similar to FZ, F3, and F4. So I'm not actually going after a microcircuit, but those are the next steps, is to fine tune the circuit.
Right. So I got a question as well.
Sure.
Because you said that the best predictor of, let's say, successful aging is this internal energy. That if you just feel low on energy, it's not so good after 10 years after your retirement, if I understand correctly. But are we doomed? Means I feel low energy and that's it. It's my destination. Or you can increase this internal feeling of energy, like forcing yourself to sleep, even if you cannot, or exercise or do anything that increase this internal and then just, reverse your fate. What do you think about this?
Yeah, I think that's good. I think that's the future. And it's not energy during the day. It's also less energy at night. It's properly sleeping so that you can be more active. Yeah, there's also these things. There's a strong covariate for aging, but yeah, I think there is a cheat code for aging.
Good. I think that's not might be connected with USVs, but that's a really good take-home message. All right. Thank you very much.